larger benthic forams too, so it's appropriate for our Hawaii celebration today. This is a talk I gave at Forams 2018 in Edinburgh in June, and because I didn't have to explain too much about forams to the people there, as far as background information, I have added a little bit of background information. Now, I've got, okay, is that, oh. I've got the
page turner is here.
The page turner's here. We've got two different things here. That's good. Okay, so.
There's a pointer also here.
Yeah, there's a pointer also here, but this is safer, yeah. But anyway, so why does anybody care about bioherms produced by larger forams? Here's one from Tunisia that I got to visit some years back, and actually was when I thought about this particular topic for the first time, or really thought about it. And so here's a question. Here's a test question for you. What are the three most valuable resources that we extract from the earth? And I'll tell you right offhand, I'm not referring to either oxygen or soil nutrients. So I'll give you a hint. What do you think are the three most valuable things we extract from the earth? Pardon? Petroleum?
Metals.
Pardon?
Metals.
Nope.
No.
Number one, actually, you're missing number one. Many of you have it in front of you. Water. Yeah, coffee. No. Yep. So yeah, water. The number three. Anybody want to guess what number three is?
Forams.
Yep. Forams contribute to it. And that's what I'm going to tell you about. But the number three, they're actually selling it on the black market. It's in India now, and China has used more of it in the last couple of years than we have in the last few decades. Does anybody want to guess now?
Concrete.
Well, earth materials for building. So water, hydrocarbons, and building materials. Okay, so why am I mentioning that? Well, there's something called the Neotethys Ocean, which is basically this, but kind of also extends because of plate movements into Australia. So anyway, the carbonate banks across the Neotethian region are valuable for three things. For example, 50% roughly of Southeast Asian and Australian hydrocarbons come out of Paleogene and Miocene. So Paleocene, Eocene, Oligocene, Miocene carbonates and other rocks, but out of their rocks, and carbonates have been studied. Actually, Shell and Royal Dutch Shell was because of their Indonesia connections. We're working out there. Okay, shallower subsurface resources. Of course, we're not interested at all in this one. And that, I used to have a nice piece of rock that I used as a doorstop, and somewhere in the course of my career, it's disappeared, but it had poker chip-sized larger forams in it from the Ocala limestone. And this is actually a thin section of a foram, and the picture is showing the microporosity of the Ocala limestone in that particular location that they were studying. Okay, so what surface resources occur in sediments and bioherms produced by larger benthic forams? Well, building material has been used for a long time by humans. and I don't know if you've ever heard the stories about the nummulites that they thought that I think Aristotle or somebody thought that they were ancient lentils that were the petrified lentils that the workers used because there were so many of these little forams laying, or not little, big forams laying around, and these are all individual forams that you're seeing in this figure. And one of my favorites, and I wish I had purchased one like it, these are larger forams and the smaller versions of the forams. So, and I'll tell you about their life cycles in a minute. But that's a tile from the one that I was looking at, I think, was at tons of tile. But you can find beautiful foraminiferal limestone tiles at your local, whether it's Home Depot or wherever you are interested in home improvement. Okay, so what carbonate banks are characterized by? Not just large benthic forams. 25% of Hawaii's beach sand, for example, historically at least, came from larger forams, but not these really big, large, flat things. Oh, my scale bar somehow disappeared. But anyway, these are on the order of a couple of centimeters at least across. Well, you've got a pencil here, and you can see the flat plates. And here you've got the big ones. This is a point of a rock hammer, and the smaller ones. And I'll tell you why there's larger and smaller ones. And this is just some examples from Italy, Spain, Germany, and actually Australia. And so really throughout the Tethyan region. So what are they? The ones that, the special kinds, that nummulite banks were the things that people have talked about and it's most prevalent in the literature, but they don't all have to be nummulites that produce these banks. They can be, but they're dominated by one or two species. They're visually, although not necessarily numerically prominent of the very large shells, and they're typically bimodal in there. So there's the half a centimeter size and the poker chip size, and they're generally low diversity overall, not just two species of one or two species of larger forams, usually just one. Okay, so early characterizations of the nummulite banks, where they were recognized from Eocene limestones in North Africa, and Libya's, some of Libya's, for example's major oil resources, Tunisia and others, are from this age of carbonate banks. And so anyway, a fellow by the name of Arne back in 1965 proposed that they were environmentally controlled where these banks would occur, that they formed in place, that they were relatively deep and often associated with coral and algae. Again, this is one from a diagram from the Sirte Basin in Libya, and this doesn't really represent the depths of these particularly well, but they were typically recognized as being offshore. Eric Egner and his dissertation work in the early 80s said, no, they're not formed in place. There's a lot of things that should, he thought that the smaller forams were removed by hydrodynamic sorting, and he provided evidence for post-mortem transport, or at least suggesting that they could have been subject to assorted amounts of winnowing. Okay, so what did I have to contribute to this long-standing discussion? Well, the previous evidence for in situ accumulation, you can't convince me that that got transported very far or had too much winnowing. But in some cases, there is evidence for limited winnowing and transport. The other thing, of course, is the fact that it's pretty much oftentimes a lot of very large individuals. And so what happened to the smaller ones? And in seeing another paper recently got me thinking about the Foram Biology and ecology and some oceanographic discussions or modeling with some carbonate colleagues. And so I realized that, you know, as geologists, we're commonly assembling elephants. Meaning, if you don't know this story, it's the blind men and the elephant, and they didn't know what an elephant looked like, and so they got an elephant handler to let them see an elephant, and each one of them got a different part of the elephant, and so each of them, one thought it was like a snake, that he got the trunk, one thought it was like a tree, one thought it was like a wall, and one thought it was like a snake because he got the tail. And so the question is how can everybody be right? And so obviously we're often looking at major differences in habitats, differences in species, and differences in life stages. So what can our living studies of our living forams tell us about benthic forams, and that can provide insight into these early Cenozoic carbonate banks? Well, the first thing is what we know about the biology and their reproduction now, and what we know about their dependence on algal symbiosis. And so the first thing is that we have the forams show alternation of generations. And those big monster forams in the bioherms are actually produced sexually. They recruited from gametes, and interestingly enough, they start with very tiny first chambers. They grow typically to much larger sizes than the asexually producing versions. Now, you can have asexual reproduction that produces schizonts that produce more, that do repeated asexual, or what they call a gamont that produces the gametes that then are typically broadcast, and the propagules recruit to form these, or to develop these big microspherics. Okay, this is just the story of the terminology that we use for them. And so the agamont, or the sexually produced versus the sexually reproducing, if you will, individuals. Okay, and in a typical population, for example, my Hawaiian studies, and actually until 1991, we had only seen this strict alternation of generations in the genus that I've worked most on. On the other hand, I used to send, actually the species that's shown here, to a fellow in Germany in thermos bottles by mail, which is also part of a little bit part of the story. But he thought he had a different species from shallow water because it would successively asexually reproduce, whereas the ones that I sent in from Hawaii that were from deeper water always showed the alternation of generations. Okay. My friend Susan Goldstein, in a very important paper in 1997, demonstrated that gametogenesis and the ability to gamete broadcast was kind of a basal reproductive pattern in forams. Then, and what this is Susan, and this is Elizabeth Alva from Norway, and they discovered that we had never seen essentially gamete fallout. The people, the 5 or 10 people who'd worked on biology of forams had never seen gamete fallout. And what Elizabeth had this brainstorm of taking samples from deep water and raising them in different environmental conditions, and darned if she didn't grow microspheric or sexually produced individuals. She grew shallow water forms from deeper water sediments, meaning that the propagules are being distributed, and they seem to be cryptobiotic, what we call cryptobiotic or dormant. She's not only taken the samples and directly, but she's left the samples in cold storage for 2 or 3 years, and then grown the forams out of the sediments and gotten the same kinds of results. So we now know this has just completely turned foram biology, foram ecology on its head. For example, I was at the forams meeting. One of the molecular people was reporting a worldwide collaboration, or he was collaborating on worldwide sampling of plankton to see what in the biodiversity studies. And he found that, or he reported that 85% of the barcodes or the molecular results from open ocean planktic samples, 85% of them were benthic DNA. So, yeah. We now know why forams can be pretty much anywhere. Algal symbiosis, widely recognized now. I was on the ground floor on this issue, and not only are the forams, these what we typically call larger forams, light dependent because of their symbiosis, but their shell shapes are very different based on where they're living in the water column. Basically, if there's not much light and it's all coming from straight above, they're flat. And if there's more light and more water motion, then they tend to be more rotund. And there's some difference between this group, who has less transparent shells than these guys. Okay, so, and the domulites tend to be some of the flattest ones out there. Okay, so a little physical oceanography here, or optical oceanography, just reminding you that, light gets a whole lot deeper in clear open ocean water than in coastal water. And here in Tampa Bay, it's probably the skin on the top of this is the light penetration. Okay, so this is an old figure from some of my early work, and just modeling how the light gets to the bottom, or how much light gets to the bottom, and then with these nice figures superimposed on that. And so obviously the clearer the water, the less food. And so what have we learned so far that might tell us something about these carbonate biostromes and bioherms characterized by larger benthic forams? Well, for example, if you've got the forams living here along a shelf, and I am not artistic, my my only art medium is bread. I make, but other than that, my brother and sister, who were artists, got all the art genes in my family, so you understand why. Anyway, so I've had a little drown platform here below any significant light penetration, and we've got the largest forams here, living right, at the deepest part of the oligophotic, as we call it, zone. I've just said euphotic here, but I should have said oligophotic, or the lowest of the light zones. Okay, but these propagules, and let me tell you that they're far smaller than this. I mean, they're like on the order of 20 microns or so. So they're not really this big, but I've kind of color-coded them such that the light ones are the shallower waters and the yellow ones are the deeper water, the nummulites. And so, okay, what happens if sea level drops or water clarity improves and the platform top is now in the deepest part of the euphotic zone? Well, those couple of species of deepest dwelling forams can recruit there. And they recruit from the propagules. And a little plug here for Ben Ross, who will be hopefully telling you more about this in his defense in the not too distant future. He really documented dormancy in foraminifera, and how it's basic to most groups of forams, at least, or it's found in all of the basal groups, I should say. And he's recently in his I can't remember if it's a paper just submitted or just out in Peer J, or if it's the one just submitted. But anyway, he's shown that they can survive 20 months in the dark, so almost 2 years. And the only reason that he stopped at 20 months is because he ran out of forams. And so anyway, we know that they can just shut down. Not just the dormant propagules, but the adults can shut down for as much as a couple of years. And so if you have seasonal or interannual conditions that reduce the euphotic zone, and one of the things about these larger forams is that they lived at much higher latitudes when the oceans were warmer during the